Okay, good morning, everybody. Why don't we get started? Okay, a couple of announcements. First, I wanted to update you on the James Franco event. This is an interview that Okay. So I told you at the beginning of the semester that I had proposed to James Franco, who's a faculty member here, to do a presentation for all NYU students called Understanding the Monkey Mind, a conversation with James Franco and Wendy Suzuki in response to his movie Rise of the Planet of the Apes. I'm sure he's fascinated with the monkey mind and all those things associated with it. But he agreed. He thought it was a great idea. And so we had wanted to try and get a date for this semester, but it didn't turn out because he's not going to be in New York very much this semester. But I got a promise from his booker that we would find a date in the spring. So watch out for the announcements. It's going to be a university-wide event, so hopefully there'll be enough seats for everybody. So that's for next semester. Next news, not so happy. Next Wednesday is our second midterm exam. And this week in lab, there is a lab this week, no lab quiz, but you're going to have another whole session devoted to review for next Wednesday's exam. There's also going to be a review session tomorrow, an extra review session tomorrow night from 6 o'clock to 7:30, and where will this review session be? 815 Meyer Building on 4 Washington Place. For all of you that went to the last one, it's the same room, same building, just down the street this way. Okay, same format as the last exam. Expect true, false, multiple choice, fill in the blank, and a couple of different kinds of short answer questions, kind of short answer questions and medium short answer questions where we'll ask you to describe these answers in 3 or 4 sentences or maybe even a full paragraph. So same kind of format. New topic, of course, we focused on sensory systems, motor systems. Oh, this is very important. We will cover all exams, including next Monday's exam. So we'll finish the motor system today. On Wednesday, we're going to go over motor diseases. That will be covered. Next Monday, will be the first lecture in the learning and memory part of the course, and that lecture will be covered on the exam, okay? So everything up to Wednesday, next Wednesday, will be covered on the exam. Any questions about that? No? Okay, great. So the other thing that I wanted to just emphasize to you is to point out to you something that you've probably all already realized, and that is the difference between what's stated in your book that you're all using as a reference for this class, and what real neuroscientists study, okay? So if you take the book, it's like a Bible of neuroscience. These are all the facts that we know about neuroscience. And a lot of the facts you've learned. You've learned about facts about neuroanatomy, names of structures, where they are, the facts of the pathways of the visual system, the somatosensory system, the facts of how action potentials are propagated. Those are all facts. But what neuroscientists like myself do is we're actually asking the new questions. And I want to make sure that you are aware of what those questions really are. So for example, let's take the visual system. That is the sensory system that we know the most about. You learn so many different facts about the sensory systems, the pathways, the rods and cones. You might think, okay, I have to learn all this. There's nothing more to learn. But just to give you a flavor, just recently, in fact, this year, at this year's Society for Neuroscience meeting that's happening starting November 10th, there is a woman who won a big prize called the Lindsley Prize for the best thesis, dissertation thesis in behavioral neuroscience. And she won this prize for identifying a whole new visual pathway, a whole new set of retinal ganglion cells. There are not a lot of them, not nearly as many of them as rods and cones, but it turns out there's a specialized subset that are important for what's called photoentrainment, that is being able to detect whether it's light or dark and modulating your behavioral state. Is it nighttime? Should I be sleeping? Is it bright out? Should I be active? And there is a specialized pathway that is not important for visualizing your face compared to your face, or spatial relationships in the visual world, but is only important for bringing light information in to the parts of the brain important for determining your general patterns of kind of biorhythm functions. Whole new pathway. Completely new discovery. She did not do it on her own. There was a whole lab and team, but she wrote a beautiful, beautiful thesis on this. Completely new pathway. And so it's not just like, oh, we know everything about the visual system. We're still learning things about something as well studied as the visual system. And what we're going to be talking about today is the motor system. Even more unknown, because when we talked about sensory systems, this is just kind of sensory passive information coming in, and we talked about the fact that we're not seeing everything out there. We're seeing a compilation of things. We are filtering things. We're filtering things in our auditory system. So our bat would hear different things than we would hear. An eagle sees a different scene than we see. Why? Because he has such more precise vision than we do. It would be a revelation to see what an eagle sees. It's very, very different. But still, relatively passive. These things are coming in. Now we're starting to look at the motor system. This is not a sensory information. This is not sensory information coming in. This is something generated by us. I can decide to pick up this pointer, press the button, and get my little red dot up there. How does my brain do this? This is free will we're talking about. We're talking about the brain areas that we are controlling to do what we want, okay? So if everybody, just for a second, everybody sit up really, really straight. Oh, okay. Everybody can do that. Shoulders back, sitting up. Everybody has the free will to do that, okay? That was your decision. What we're going to be talking about today is exactly what happens in your brain when you do that. Do I know what free will is? From a neuroscience perspective, I don't. But we're going to talk about the pathways that we do know about how you are all able just to do that. Just to do a simple thing of sitting up straight in your chair. Okay, so we're going to continue on where we left off last time. We talked about all the muscles in your body. Everybody knows what muscles are, and you just stimulated a whole bunch of them, particularly in your core, in your abs, in your back, to do that sitting up straight thing. Those are slow twitch muscles. We talked about fast twitch and slow twitch. Those muscles that help maintain our posture are the ones that continue, that can fire for a long, long time. And even those get tired, as we know, as we get tired in a lecture and we start to slouch and we start to move back in our chairs. But all of those muscles are the ones that you just activated here. Each of the muscles are made up of lots of individual muscle fibers. And the individual muscle fibers that we talked about are made up of two key elements that you need to know about. Actin, which is the thinner element, the blue strands here, and myosin, the thicker element, which is the thick thing in the middle. And muscles, what they do is contract. And so what happens is the myosin hooks on to the actin and pulls it together. And that's how a muscle like the bicep can contract, and you can flex your arm with the contraction of the bicep. Okay, we talked about fast twitch muscles, slow twitch muscles. The prototypical fast twitch muscle was your eye movement, your eye muscles, controlled by cranial nerves, okay? And so those are very fast, they're very precise, but they don't have to lift a heavy load. Your eye is very light, and they don't have to be lifting things like big books, okay? Slow twitch muscles are not nearly as fast and precise and can turn on a dime or can contract on a dime, but they are more resistant to fatigue. Those are the muscles that are helping you maintain your posture, your abdominal muscles, your back muscles. Another good analogy is the muscles that sprinters use. So we talked about the fact that dark meat in in the chicken that you eat is a lot of them are slow twitch muscles. These are the ones that are maintained for a long time. But within the legs, there are, in fact, both fast twitch muscles and slow twitch muscles. The fast twitch muscles are those muscles that help the runner kick off at the beginning of the race. When that gun sounds, those fast twitch muscles are the ones that contract rapidly and on a dime. But it's the slow twitch muscles that gets the runner through the main part of the course. So again, both of these are acting together. Okay, so what is actually controlling the muscles? And that's where we get into the central nervous system, to special muscles called motor neurons. In fact, we're going to be focusing on, we're actually going to be talking about two types of motor neurons. One is the alpha motor neuron, and those are the neurons that are controlling all our striated muscles, our voluntary muscles. So for example, the heart and the stomach, those are muscles, but those are smooth muscles. Those are controlled by unconscious autonomic and the autonomic nervous system. Striated muscles, biceps, triceps, quadriceps, hamstrings are all striated muscles and controlled by alpha motor neurons. Where are the alpha motor neurons? Alpha motor neurons live in the ventral horn of the spinal cord. So we're going back to the spinal cord and hitting kind of the second part of the spinal cord. Let me just jump here. Here is our nice spinal cord again. Here is the dorsal horn of the spinal cord, and here's the ventral horn. The ventral horn right here, this gray matter where cell bodies are, these are where the primary alpha motor neurons live. Those neurons are sending those axons out this route. It's the ventral route of the spinal cord. Remember the dorsal route, what comes through the dorsal route? Sensory information, right. And what is sitting right here in this dorsal root ganglion? What's in there? Cell bodies. Cell bodies of what? Of neurons. What kind of neurons are they? What are they doing? This was the sensory part. So remember, these are the cell bodies of those neurons that are going out and sensing all of the sensory information from our body. Remember the Pacinian corpuscles, Ruffini's corpuscles, those are connecting to the afferent input of these primary sensory neurons in the dorsal root ganglion, and the axons of these neurons here are going through the dorsal root and then going up into those sensory pathways. That's half of the spinal cord, the sensory half. Now we're finally getting to the ventral half, the motor half, where we have the cell bodies of the alpha motor neurons sitting right here and making up this ventral horn of the spinal cord. Again, these neurons, their axons are going out towards the ventral root, and then they come together. So the spinal nerve, remember, the 31 pairs of spinal nerves. Now we're going back to the very beginning of our lectures. Those 31 pairs of spinal nerves going all the way up and down your spine are made up of both sensory inputs from the periphery, whose cell bodies are in the dorsal root ganglion, this lump right here, and the motor neuron axons that are going out to stimulate the neurons, okay? So what happens when you cut a spinal nerve? What would happen if you cut this spinal nerve and it's innervating the arms? Let's say one pair of spinal neuron innervates the arms. What would happen if you cut this? You would be paralyzed. What else? So you couldn't move because you're cutting the motor neurons, but what else? You couldn't sense anything. So you lose all sensory and motor actions for this part of the body that this nerve is innervating, okay? Both sensory and motor, okay? And so it's it's that way because you have both sensory inputs coming in and motor neurons going out this one individual nerve. So that's why you don't want to have nerve damage in your spinal cord. You have motor problems and you have sensory problems. Now, if I only went in and lesioned the dorsal root ganglion cells, then I would have purely sensory problems. If I cut the dorsal roots right here, I would only have motor problems. Okay? So you should have a 3D understanding of this spinal cord because we've gone over it at multiple different points during the class, and you should be able to understand that or predict that if I damage the cells here, what would happen? If I cut the spinal cord here, what would happen? If I cut the nerves here, what would happen? What would be the prediction? Does everybody get that? Does everybody have a clear idea of what would happen in those different situations? Yes, question.
So the primary alpha motor neurons are in the ventral horn
of the spinal cord. And the dorsal part of the spinal cord is primarily sensory, okay? That's where the sensory pathways are coming. Sensory neurons that are helping to convey sensory information are in this dorsal part of the horn. And the ventral part of the spinal cord, the ventral horn and the ventral part of the spinal cord are dedicated to motor functions. Any other questions? Okay, great. So motor neurons, so those motor neurons are like any other neurons. They generate action potentials. And the action potentials in these alpha motor neurons travel down the motor neuron that branches into many terminals near its target. Now, this target of a motor neuron is a muscle, okay? It's not another neuron. It is going to terminate in a muscle. And it's action potentials from the motor neuron that allows your muscles to contract. So my neurons probably in the mid of my back are contracting as I am as I'm contracting my bicep muscle. And the neurotransmitter that this motor neuron uses at the level of the muscle is acetylcholine, okay? So here is a blow-up of an alpha motor neuron. It's the cell body. And it's just to show how many different inputs there are to the motor neuron. The axon is coming out here. It has lots of dendrites. And we're going to talk about the major input to this motor neuron in a second. What controls this motor neuron that goes out to actually contract a muscle? Okay. So, but first, before I do that, I want to talk about the actual transmission of the action potentials from the motor neuron to the muscles. And that happens at what's called the neuromuscular junction. And that is where the motor neuron terminals and the muscle fiber meet. Okay? This is a synapse like any other synapse, except the postsynaptic part is not another neuron. It happens to be a muscle. And the effect of acetylcholine is to actually help that muscle contract. That actin and myosin is sliding up against each other and contracting because there's acetylcholine in the vicinity. Neuromuscular junction is a very, very effective synapse because almost every single action potential elicits a contraction. And an important concept is the concept of a motor unit. A motor unit is a single motor neuron's axon in all of its target fibers. So a motor unit are all the muscles that a single motor neuron is contracting. Okay? And that can be seen as analogous to something we talked about in the sensory system, in the somatosensory system. Remember what I talked about? A dermatome is a part of a skin innervated by a particular spinal nerve. So we're not talking about individual sensory neurons, but a dermatome is, like here is the arm dermatome. One of the spinal nerves innervates this entire part of the arm, and its cutting of this particular spinal nerve will not only cut off all sensory feeling to this arm, the dermatome of this nerve, but also all the motor input. So the sensory and motor kind of flow in a specific spinal nerve is matched. They're going to the same places. Okay, so here is this picture again, and now what we're seeing is a spinal nerve coming out, and we're seeing the axon of a single neuron within the spinal nerve. There's multiple alpha motor neurons going out each individual spinal nerve. It comes down and you can see the termination in the muscles. Here's this person's bicep muscle, and here is, again, these are myelinated axons. We want, obviously, fast responses. These are myelinated, fast responding axons, so that if you're a ping pong player or a tennis player, you can have very, very fast responses, or a football player, as we saw at the beginning of the last lecture. And here is the myelination terminating, and here is the neuromuscular junction, the termination of the axon onto these striated muscle fibers. Okay, and here it is in a larger blow-up. Here is the actin and myosin, and here is the axon terminal, and this is where acetylcholine is released in the same way with vesicles containing acetylcholine. The vesicles release, and acetylcholine then contacts the muscle fiber that makes it contract. Okay. We call, just a term that you should know, the alpha motor neurons in the ventral horn of the spinal cord are what's called the final common pathway through which the brain and spinal cord controls muscles. So let me just clarify something. You remember, I think it was the very first lecture. I told you to close your eyes for a second and wiggle your right pinky toe, right? And I said there was this long pathway of a single axon that goes all the way down. It's not really a single axon. What's really happening there is, by wiggling my little toe, I have neurons in my little toe area of my primary motor cortex. Those are wiggling. Those are firing action potentials. Your brain isn't wiggling. Your toe is wiggling. Your brain in those neurons in primary motor cortex, in the cortex, are firing. Those axons are long. They're not quite as long as going all the way down to your toe, but what they do is they go from the top of your head probably down to the bottom of your back. Okay, so that's still a very long motor neuron from primary motor cortex. Those primary motor cortex neurons synapse on the alpha motor neurons in the ventral horn at the base of your spine, and those motor neurons then go out and terminate in the muscles that are controlling the movement of your baby toe, and that is a very, very long pathway, perhaps not as long as I indicated at the very first lecture, but still quite long. And imagine that for a giraffe. Okay, still hugely long neurons we're talking about in the motor cortex and motor pathway. Okay, so now we get to a really important concept in motor function. It's not just all about the efferents going out. So efferents are the signals from the central nervous system out to the muscles. That would be an efferent. An afferent signal is information coming from the outside in. So sensory information is afferent information coming from the periphery towards the central nervous system. You need to know the difference between an afferent and an efferent of the brain. So in the motor system, we're talking about efferent signals, and we think that's the main thing. We have to get that signal from the brain to the muscle so we know where to move. But it turns out that even the motor system not only has efferents, but has important afferents, which is called, underlies our ability of what we call proprioception, with the collection of information about body movements and positions. It's not all about commanding what we're going to do, but it's also about paying attention to where we are so that we can monitor what's happening and adjust if necessary. And we're going to talk about two different kinds of proprioceptive mechanisms we have in the motor system: muscle spindles and Golgi tendon organs. Okay, so first, muscle spindles. Where are these? Muscle spindles consist of afferent and efferent elements. Intrafusal fibers lie within the spindles. Let me just show you a picture of this. Okay, so this is a picture from the book. It's a really nice illustration. Here is your bicep muscle, and within the bicep, let me just identify, here's the muscle itself. Here is the tendon. The tendon connects muscles to bones, okay? So if you have a torn tendon, that's really bad, but that is what's connecting your muscle to your bone. Now, for the muscle spindles, the muscle spindles are within the muscle itself. And each muscle has two major types of fibers. Extrafusal muscle fibers, which are basically the regular muscle fibers, the striated muscle fibers, and the intrafusal muscle fibers. These are the specialized muscle fibers that make up the muscle spindle. And they're kind of inside the regular muscle, and they're kind of a cylindrical shape. So it's basically a muscle spindle is a specialized kind of muscle fiber. It's called an intrafusal muscle fiber. And this muscle fiber has sensory information that is obtained from this muscle fiber. There's two types of sensory endings. One that terminates on the middle of the intrafusal muscle fiber right here, and one that terminates on the end. These are called secondary sensory endings that terminate on the edges, and these are primary sensory endings. What these sensory endings do is they sense stretch of the muscle. So let's say my muscle is here, hanging out, my bicep muscle is here, hanging out, not doing anything, just hanging out, and I have a big book. Somebody hands me a big book, and my bicep muscle stretches. That stretch is what these afferent fibers are very sensitive to, and you'll get activation. At the beginning of the stretch, you'll get primary activation of the primary sensory endings. These primary sensory endings are very sensitive to the dynamics of new stretches of the muscles. And then the secondary sensory endings, those are more sensitive to continuous stretch. So if the stretch continues for a long time, if I continue to hold this book here and nobody helps me do something with it, then that's when my secondary sensory endings kick in. Okay? And so what this intrafusal muscle fiber does, what this muscle spindle mechanism does, is that it detects stretches of muscles, and it can then inform the rest of the motor system to adjust accordingly. So we want to know when our muscles are being stretched and when they're not, and these extrafusal muscles are exactly what's doing it. Now, so let's look at this for a second. So here is the extrafusal muscle, and we've plucked out the intrafusal muscle, or the muscle spindle, right here. Here the muscle is relaxed, okay? And there's not much activity. Just focus here on this top row, the muscle spindle activity. And here what we're monitoring, each one of these dots is an action potential. Let's say it's from the, our primary sensory ending, okay? So these are action potentials from the primary sensory ending. The muscle length is just normal, but then we stretch the muscle. Something stretches the muscle stretches, and then you get lots of activity in the muscle spindle, telling us that the spindle has been activated. And then what usually happens in other motor systems is the muscle then contracts to counteract that stretch. And then you get lower activity in the muscle spindle. But look what happened here. This muscle is now significantly shorter than it used to be because it's stretched. And then we have a muscle spindle that's the same length. So it's kind of hanging out there. It's way too long. And it's not sensitive anymore to stretches even of this level of contraction. Okay? And so that's where the muscle efferents come in. So here, each muscle spindle doesn't only have primary and secondary sensory endings that innervate it, but it also has what's called a gamma motor fiber. A gamma motor fiber, not an alpha motor fiber, but a gamma motor fiber that comes in. And what this does is it can control the length of the actual muscle spindle. So let's say you have a stretch and you contract your muscle. Well, the gamma motor fiber goes back and contracts this muscle spindle to match the length of the rest of the extrafusal muscle fibers, so that this stretch receptor is now sensitive to whatever length your muscle is. Okay, does that make sense? It's just going in and realigning the intrafusal muscle fiber to the rest of the muscle. Okay. And let's see. Right, so we talked about dynamic stretch is what the primary endings are sensitive to, and static stretch is what the secondary endings are sensitive to, and we talked about gamma motor neurons. Now, the second kind of proprioceptive mechanism that we have is called the Golgi tendon organ. The Golgi tendon organ is not within the muscle itself, but it actually sits, these are fibers that sit on the tendon that is connecting the muscle to the bone. Okay? So these are simply sensory endings here that's monitoring the tension that you see at the level of the tendon. Okay? So this is really a safety valve. If there's too much tension there, that is, you have a really, really hard load that's actually pulling on the tendon that is connecting the muscle to the bone, you have the Golgi tendon organ, which is a safety valve. So let's see what happens here. We have we have a stretch here, and when the muscle is stretched, and this is stretched a lot, you not only get activation of the muscle spindle, but you get a stimulation of the tendon as well. And so at this level, you get Golgi tendon organ activity. And what the tendon, this is actually only activated in more vigorous stretching, when there's actually tension put on the tendon. And so what happens there is, once the Golgi tendon organ is activated, you get signals back to the muscle that release the stretch of the muscle, and actually releases tension on the muscle. So it's, again, a safety mechanism to try and safeguard your tendons so that they don't pop off of your bones. Okay, so that is Golgi tendon organ. And one nice way to summarize what's happening, particularly for the muscle spindles, is to go back to a activity or an example that we're very, very familiar with, and that is the stretch reflex. Do you remember at the beginning, at the end of our action potential lecture, we used the example of a doctor hitting your knee and giving you your kicking reflex, right? And same thing, this is another example of that. This is a stretch reflex that we're going to talk about. And this is a stretch reflex that is only happening within, between the muscles and the spinal cord. The primary motor cortex is not involved at all. So what happens here? So again, I'll translate this for our hitting the knee example. So what happens is the bicep is at a normal length right here, and you put a weight in the person's hand, and that stretches the hand. You know, outside force stretches the hand. And then these bicep muscles are stimulated. So stretching of the muscle, you get stimulation of the muscle spindle response, and that goes in through. These muscle spindles also have their cell bodies in the dorsal root ganglion. All sensory neurons are in here, really important ganglion right here. These axons of these sensory neurons go and they synapse on the motor neurons that control this muscle. And so what this stretch reflex is doing is this monosynaptically, sensory input is coming in, going to a stretch. This says, okay, I want to contract this muscle that's being stretched. So then you have a motor command going out and stretching, and contracting the bicep muscle. But you can't, everybody just contract your bicep right now? Okay, is that the only thing you're doing? Can anybody only contract their bicep, and that's the only muscle that they contract? Is that possible? No. Why is it not possible? Yes? Exactly. Perfect. Muscles come in pairs that are like opposites. We talked about antagonistic muscles. What is an antagonistic muscle for your bicep? Tricep, right. So when you contract your bicep, you're stretching your tricep. And so when you do this exercise in the gym, you're contracting your triceps. And so these are antagonistic muscles. So in a system like that, you can't just stimulate your or contract your bicep. This second neuron here is going to inhibit. This purple neuron here is an inhibitory interneuron that is now going to inhibit the neurons of, sorry, not to inhibit, but it relaxes the muscles of the triceps, okay? So this reflex is a stretch is detected. You get stimulation of some muscles that causes a contraction of the bicep and a relaxation of the tricep. That's exactly the exact same pattern that we talked about in the beginning of class when we were talking about action potentials, chemical and sensory action potentials. The same thing was happening. What happens when we hit the knee? You get a stretch. You actually cause a stretch of the muscles in the quadriceps that cause the same loop to happen. But now you are contracting the quadriceps and you're relaxing the hamstrings. Okay, so this is a nice example of a motor kind of spinal reflex that now you know how exactly these muscle spindles work in there. So you think, oh, I've never heard of muscle spindles. They can't be that important. Well, that's what a very important physiologist, Sherrington, thought as he was studying the sensory and motor system. So he decided to cut off all of the afferent input, that is the proprioceptive input from the muscles back to the spinal cord. So these muscles had perfectly working efferent signals. The acetylcholine was working, they could stimulate something, but he only cut these muscle spindles and Golgi tendon organs. So what do you think would happen if you did that? Anything? Would the animal, or would you still be able to move around just fine if you didn't have input? Yes?
The brain wouldn't really be able to tell what the muscle was doing.
Right. Exactly.
So you wouldn't have any understanding of what your
Exactly. Your brain would have no understanding of what your muscles were doing. And so what would happen is when they would cut off all the afferent input from the motor neurons, let's say from the right arm could the acetylcholine, the alpha motor neurons were going perfectly well to the muscles, but with no afferent input, the animal stopped using that arm. It just went limp. Didn't know what to do with it. Couldn't do it until they restrained the other arm and started training the animal. And with time and effort, the animal was able to slowly use the arm that had been de-afferented. But that just shows you how important these afferent inputs are. It's not all about the signals to move your muscles, but it's about the proprioceptive information about what your muscles are doing that allows you to adjust and to continue to move. It's all done unconsciously, really. That is the proprioceptive part, but it's critical to our ability to move and to learn very complex motor actions. So that's why we spend so much time talking about both the muscle spindles and just two examples. Proprioception is much more complicated than that. We're really giving you two simple examples of how proprioception works. Yes?
So if you cut off the efferent signals, then you can't move or feel anything, but if you cut off the afferent, then you can't feel, but you can still be trained to move.
Yeah, if you cut off the efferent signals, the alpha motor neurons, you cannot move at all. There's not a physical way to get the acetylcholine to the muscles. You can feel in the sense that you have the spindle afferents coming back, but you can't do anything with them because what that does is it just helps you move better, right? But exactly as you said, if you cut the afferents from the muscle spindles but leave the efferents, you can still move, but you have to be retrained to move because this is so integrated, both the afferents and efferents in our motor system. Okay, does that make sense? Any other questions about that? Okay, good. Okay, and then so those examples are focused on the major striated muscle groups of your whole body, the muscles that allow you to walk, to run, to play the piano, to do everything like that. But there are, as we learned, a whole group of muscles that are controlled by cranial nerves. And that means that it's not, the motor neurons are not in your spine. They're actually in your brain stem. So this is what the book calls movements are controlled. So some muscles are controlled directly by the brain. And you already know these muscles. You know them as the cranial nerves that have motor functions. And these motor functions are all related to the head and neck, including sternocleidomastoid muscle in the back of the neck. All of the eye movement muscles, which are the fast twitch muscles, are controlled by very specific neurons in the different cranial nerves. And so you should re-familiarize yourself with the cranial nerves and which ones are motor, particularly the motor neurons important for eye movement. Oh, one last thing that I forgot to go over is, let's see. This, I really like this concept, the innervation ratio, okay? The innervation ratio refers to the number of fibers, muscle fibers, innervated by an axon, okay? By a single axon. So, compare and contrast. Let's say I have an axon that innervates 500 muscle fibers versus an axon that innervates 3 muscle fibers. What do you think the difference between those two things would be? What would I use the axon that's innervating 500 muscles versus the axon that's innervating only 2 or 3 muscle fibers? What would be the difference there? Yes?
The one innervating a smaller amount is fast twitch.
It could be fast twitch, but what do you think is the nature? You're very close. What is the nature of those muscles? The muscles that have just one axon innervating just a few muscle fibers. Yes. They could be smaller. Yes, back there. Sorry? Finer. Yes, finer movement. Exactly. So. Yes, right, exactly. So, exactly, it's the same concept. He was referring to the concept of convergence versus divergence. And, but what you said first was exactly what I was going for. You have to think about the finesse of the movements, okay? So try and write with your hand, and then try and pick up a pen with your 2 elbows and write. What are you better at? Writing with your hand, right? So what's the difference between motor control of your hand and motor control of your elbow? Yes? Right. It's lower innervation ratio. So you want a small number of muscles to be innervated by a single neuron. And so it's just think about all the parts of your body that you have fine motor control for. And those are the ones where there is much more detailed innervation of individual neurons to small numbers of axons. So you can imagine your hands have very small innervation ratios. Your eyes have very small innervation ratios. But my quadriceps and my hamstrings have much larger innervation ratios, okay? It's not, well, maybe dancers can refine that, but the rest of us But even dancers will have better and more intricate motor control in something like their hand. Okay, so now we're to the spot that we always talk about in sensory motor pathways, sensory motor systems, I should say. What is the pathway? So we spent a lot of time comparing and contrasting the input from the somatosensory system, the visual system, the auditory system, how they're similar, how they're different, how on those sensory systems, all of those sensory inputs are going through the thalamus. Now we're going to talk about how information from primary motor cortex gets down to the alpha motor neurons and finally out to the muscles. And this information is getting out through two major systems. In the rest of this lecture, we're going to be talking about the pyramidal system or cortical spinal system, and the other system that we'll focus on next lecture, particularly in our discussions of different diseases of the motor system, is called the extrapyramidal system because it's outside of the pyramidal system. So what does the pyramidal system consist of? These are neuronal cell bodies in the cerebral cortex, primarily in primary motor cortex. They're axons, and they're axons that pass through the brainstem to the spinal cord, forming what's called the pyramidal tract. So let's see where that is. Here is primary motor cortex. Here, all these neurons in this gray area where the cell bodies are going down, and they kind of travel in one part of the upper medulla. They kind of make a little pyramid or a little lump on the medulla that we should have pointed out to you in your sheep brain dissection. You can see the pyramids of the medulla in the brain. There's basically a bump on the medulla, and that bump consists of all of the axons from primary motor cortex that are going down. At the level of the lower medulla, these axons cross over because, like the sensory systems, right motor cortex controls the left side of the body, and left motor cortex controls the right side of the body. And so that desiccation or crossing over happens at the lower medulla, and that's called the desiccation of the pyramidal tract. The pyramidal tract crosses over, goes down, and then what it does is these neurons are terminating in the ventral horn of the spinal cord at the level of the neuron that these primary motor cortex are innervating. Okay, so that is what's happening. So you're wiggling your right little toe, and we start from the left side. Let's say this is the left side of the brain, and it comes and it goes over to the right side. We go down to the lower spinal cord, way down here, and this neuron from primary motor cortex then synapses on a alpha motor neuron, or a set, I should say, of alpha motor neurons in the ventral horn. Those alpha motor neurons then go out and exit through a spinal nerve and go down and innervate those muscles that are allowing you to wiggle that right little toe. Okay, let's see. So where is primary motor cortex? We talked about this. Primary motor and primary sensory cortex are on either side of the central sulcus. And here is something that you're very familiar with, a representation of body parts in the primary motor cortex. And just like in the sensory system, there is a motor, just like a sensory homunculus, there is a motor homunculus with certain areas that are highly represented. Why? Because they have very low innervation ratios. Just more neurons are controlling certain parts of the body because it has much more intricate control of it. And those parts, you can imagine what they are: the hand. And if we look at our cheat sheet here, obviously the hands are very highly represented, and the mouth as well, because the motor actions of actually speaking are some of the most complicated motor functions that we can do. And some of us can speak very, very quickly, and so that really increases this. I should say that both the sensory homunculus and the motor homunculus are very, very highly simplified for these diagrams. It's more complicated than this is just the huge hand area. The hand area is more integrated and more intermixed with the wrist and the elbow and the rest of the arm than is indicated here. But this is a good way just to get a general feeling for what parts of the brain are controlling different motor functions. Okay. So let's talk about what exactly happens in primary motor cortex as you are voluntarily making a movement. And what has been studied in the laboratory has been reaching tasks. So animals are taught to reach or move a joystick in different ways. And in a reaching task, muscle cells change firing rate according to the direction of movement. So these cells are actually sensitive. These cells fire whenever the animal has is moving his hand in a particular direction. And each cell has one direction that elicits the highest activity. And what you can do is record not only from one cell, but a whole array of cells and get what's called a population vector. It's a population analysis of what the brain is telling the body to do. And the secret and the key is, can you decode, without knowing what the animal actually did, what he was going to do just by reading the output of neurons in primary motor cortex? So here is an example of a neuron that was recorded in primary motor cortex as an animal was doing a joystick kind of reaching task. You can see he was instructed to either move the joystick here, 90 degrees, 45, 0 degrees, down, 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 all these different 3, 6, 8 different directions. And he had a joystick, and we could tell exactly when he was making the movement. And what we're looking at here are 5 individual trials. 0 is this point here in the trial that's going from here to here. This is time on the x-axis. 0 is when his actual physical movement began. In each of these individual ticks represents a single action potential that we're recording from these individual units. So you can see that right before the animal makes the movement, the motor neurons are starting to fire, indicating or signaling a movement in a particular direction. And so we can look at the pattern of activity whenever the animal moved up. 90 degrees up this way. You can see a pretty good firing every single time. So let's compare that to when the animal moved here at 0 degrees. No activity. In fact, this neuron seemed inhibited right before this movement. But over here, you see good movement, good strong action potentials that are starting right before the movement, right before this 0 point, and continuing on through whatever, to about 250 milliseconds after the movement, but only for specific directions, okay? If you ask the animal, or if the animal moves in other directions, this neuron is not firing. Yes?
Why is it that it's only, the response is only on the left as opposed to the right?
Yeah, because that is where the neuron happens to be recorded from. So this neuron, this one neuron, it's a single neuron. So this has sensitivity to movements in this direction. But another neuron, if I record it in another location, could have sensitivity over here, some over here. And this is kind of a broad tuning. You know, what if we had more sensitive, we wanted a very specific signal? There are some neurons that only respond when the animal made a movement in this direction. So you have a whole mix of responses of these neurons, some that respond for just a specific individual direction, and some for a large range. So this is almost half of the whole, you know, pie chart here, and some don't respond at all. Yeah.
So the kind of ideal response at the top where it starts firing right before the movement,
Yeah.
you could theoretically find a neuron that has that response for every single direction on the...
Exactly. Yes, you could theoretically find a response, beautiful response like this for every single direction around, if you record around in the motor cortex. Yes, question.
So, what's happening with
Over here?
between 180 to 170, the very bottom of the response?
This one? Yeah, okay. So, yes, the firing of action potentials. For this one, as well as these, it starts a little bit before the movement and continues on after the movement. So you know, they're still doing it. They're still probably clutching the little rod there. And you can see this neuron has quite a complex response that differs in subtle ways in all these directions that it's sensitive to. So this one is mainly before the movement, right? And these continue after. Yes?
So, would there be other cells that would be firing just before the movement for the degrees at which this particular cell is firing during the movement?
Yes. Yes, there are overlap. And that's a really good question. There is this wide range of different patterns of activity that what we're trying to understand, again, is this population code. All of that together allows the animal to do his movements. He's doing what he wants to do. He's going to get a, you know, we cue the animal, go here to get a reward, and he does that, okay? And you have this wide range of cells. Some of them, they're responding to all these different things, not just when he goes up here to get a reward, but when he goes down here to get a reward. And there's subtle differences. And all of that complexity is underlying our great, you know, flexibility in all the different movements that we can do. Any other questions? This is really the first time we've gotten really deeply into kind of neural responses. Okay, so we're going to really talk about this a lot because this is a really fascinating topic. And what I'm showing here is simply a sensitivity or directional tuning curve of this particular neuron. So this neuron likes to respond primarily at 180 degrees movement, and it has good responses through a lot of it and very poor responses out here. So again, you can imagine that this is one neuron. Other neurons might have a very sharp curve like this. Other neurons may be like this. Other neurons may be just flat like this. All of these types of neurons you can see in the motor cortex. So before we go on to, let's see, do I want to do this first or do I want to do my exercise first? Oh, okay. Well, let's do this first. So imagine an animal grasping a joystick, and he's going like this, and he's moving this way and that way and that way. One question that motor neuroscientists, motor physiologists ask are individual neurons that we're recording from, are these individual neurons that we can get activity and measure their action potentials, are they coding for a particular movement, a particular set of muscles that go this, that make you, you know, go like this, or are they coding for a particular movement direction? That is, I can go like this, but I could also go like that. And I make the same kind of outward movement. And so that's exactly what they tested. They tested a monkey here, and he was looking at a monitor, and he got cued there. And with his arm, he had to move his move this apparatus to this direction right here. And he moved and rotated and he could do all these different directions. But the cool thing is that they asked him to do it in two different grips. One grip like this and one grip like this. So you can imagine that when you are moving the joystick like this, you have different muscles being activated, but you make the same kind of movements versus this. Same movement, but using very different muscles. So the question was, are these individual neurons in primary motor cortex coding for the specific muscles that are activated or for the outcome of the movement, which seems like more of a cognitive outcome? And what they found was some neurons coded for the specific muscles. That is, they differentiated between this movement and this movement. Okay, they could tell the difference. The majority of the neurons recorded did not differentiate between this and this. So that means these neurons were coding the actual outcome of the motor movement, not the specific muscles that were being encoded. okay? So I wanted to do a little exercise just to get you guys to understand what's going on and kind of make sure that you get this, okay? So you guys in the center here, you're going to be primary motor cortex, okay? This line right here, guys, are all cells that encode my movement over here, okay? Firing of you makes me go like this. You guys in the center, you guys are all going to be fire neurons that control my movement this way. And you guys over here are going to move, activity there is going to control my movement to the right, okay? So you can make me do 3 different things. You can make me go this way, or this way. This is a very simplified view, but I wanted to give you an idea of what the whole cortex is doing, not just an individual neuron, okay? So how are you going to fire? You're going to go like this, okay? So you guys fire. Okay, great. You guys fire. And you guys fire. Great. Okay. So you guys are all you're all neurons, okay? And you have specific sensitivities, okay? So make me fire so I go this way. Great. Okay. Fire over there. Okay? And fire over there. Okay, so now make me go this way. Okay, good. So now imagine I can go and record, and I can record your activity. I'm just monitoring how much you wave your hand as I'm moving my arm. Okay, so I'll get a specific pattern of activity. If I ask the animal to move his arm over there, I get no activity out of you, right? But once I ask you to move my arm over here, I see lots of activity. Or if I have the movement go through this direction, I see your activity. So that's very, very simplified. It's not nearly as simple as that because some of you are sensitive to lots of different complex things. Some of you on this side are also sensitive a little bit to my movement over here. So you have a larger kind of sensitivity. Some of you are very, very sensitive to only movement, and I have to do this in a particular way for you to fire. But the pattern as a whole is what's actually activating this set of muscles. And now we know that it's not just a set of muscles, but it's an actual motor outcome that we're signaling. That is, it doesn't matter whether the animal's arm is like this or like this. Firing of a particular neuron will be similar depending on the outcome. Yes?
Can neurons fire over there and over there, but not in the middle? And skip and go over?
Can neurons fire over? Yeah, any pattern is possible. What is your outcome that you're interested in?
Bypass, like, let's say, like the middle.
Yeah, sure. I mean, I gave the example of making a movement like that, but you can certainly go this and that. So yeah, it is kind of topographically organized, but there are individual movements that you can do. And imagine is anybody going to Fall for Dance, that thing at the city center where you can get all the tickets to the dance? I mean, imagine all the different movements. This is a modern dance. If you don't know about it, you should go. Modern dance festival every fall, and for $10 you can see all kind of the world's best choreography or dance troupes. And you go there and you see all these different movements that people do for art's sake. And yeah, it makes you realize that, yeah, you can train your motor cortex to do almost anything. And the motor cortex of those dancers are very different from our motor cortices if we're not, professional dancers. But the other cool thing is, think about you guys as a cortex for a moment. Okay, so your firing is controlling this movement right here. But it's only controlling the movement. You're in the primary motor cortex if I have my alpha motor neurons viable, right? So I have to have my alpha motor neurons or I can't move the muscles, right? So let's say you're still fine. You're still there. You still know what to do. You still kind of give the commands. But now I'm paraplegic. Okay? I can't move my motor. I can't move my legs. Or I can't move my arms anymore. this is what neuroscientists realize, that there's paraplegic patients that have damage to the spinal cord that eliminate the ability to have movement in the muscles because alpha motor neurons are done, still have primary motor cortex. You guys are still commanding these different movements. So what if we were able to actually read you out in the same way I was able to read you out? What would that be able to do? That would be able to read out what my motor cortex is telling my body to do and instead have a computer do it. So that's exactly what a neuroscientist at Brown University named John Donohue decided to work on. He was a classic physiologist, recording in primary motor cortex, recording in monkeys in tasks exactly like this, but realized that this, an implication of understanding primary motor cortex is actually I could read out your motor intentions. And if I knew what you wanted to do, even if you were shut in, even if you were paraplegic, I could actually read that out and maybe have a computer do it or a robot do it or control your wheelchair. So let's look at this video. Which describes this ability. And what you're going to see is this is one of the first paraplegic patients that was implanted with electrodes so they could actually record from large parts of her primary motor cortex, just like they were doing in the monkeys. But this time, what they're doing is taking that neural activity, trying to really decode it. And because she tells it's not a secret, you know, she says, okay, now I'm thinking about moving my right hand, my right hand, my right hand. And they figure out how to decode that means moving my right hand. And let's see what they're able to do.
E. Hutchinson is among the first humans to have her brain directly wired to a computer. Years ago, Kathy suffered a stroke that left her mentally sharp but trapped inside a paralyzed body and unable to speak, locked in, like Scott Mackler. 3 years ago, Kathy volunteered to have the same kind of sensors we saw in the monkeys implanted in her motor cortex, which controls movement and is located right on the surface of the brain. The sensors connect to the computer through this plug on her head. The system is called BrainGate, and it was created by a team led by Brown University neuroscientist John Donohue.
If you look at this square, each one of these little black boxes is the electrical signal coming from one electrode in the brain.
And each one of those is a neuron firing.
Right. It's its electrical potential. It lets out a 1,000th of a second pulse.
How well do we understand this language?
We have a somewhat of an understanding. We know that there's a general pattern of, for example, left, right, up, down, even fast or slow.
Scott, Kathy now has neural control over that cursor.
Dr. Lee Hochberg of Massachusetts General Hospital is leading the clinical trial. We watched together as Kathy showed us what she can do.
There's a handful of icons that have been placed on the screen.
Here's Google, the New York Times, the Boston Globe, and here's Mass General Hospital Stroke Service.
Yep.
We're seeing Kathy moving this cursor with nothing but her mind.
That's right. She's thinking about the movement of her hand, and she's moving the cursor much as if she had her hand on a mouse.
So if a patient who's paralyzed thinks, move my left arm, the brain fires those neurons.
Yes.
Even though the arm does not move.
Yes, it's very surprising. It fires even though you're not moving.
Cursor's still a little bit wavier some days.
Moving the cursor with her mind is not as fluid or direct as using a mouse. While we were there, the cursor meandered a bit, sometimes overshot. But Kathy always hit her target in the end.
You want to play some music? All right. She'll click on it. Imagine squeezing her hand, which is the, or doing something else for the click.
And she just clicked play.
Yep, she did.
That's pretty amazing. And so, I mean, if Kathy can control a cursor, she can control anything a computer is connected to.
That's the goal.
The lights, the temperature in the room, even a wheelchair at some point.
Ready to try it for real?
In fact, Kathy has already driven a wheelchair.
See if you can drive it right over to the door.
They haven't let her ride in it yet for her own safety. But with monkeys adopting robot arms and a completely paralyzed person driving a chair, imagine where this could be headed.
Fantastic. Very good.
Okay, so I think you can understand the implications of this. If you could read out your motor intentions, you can have robots do things, you could, and it doesn't have to be paraplegics. It can be, you know, I think about turning off my lights. I don't have to press that thing on my fancy new iPhone, and lights will turn off. So we'll continue this next on Wednesday.